the photosystems are made of a lot of different proteins, and there are these surrounding the reaction center. The reaction center is going to be where our chlorophyll molecule is, that's going to physically lose its electrons into the electron transport chain. And the reason there's this special chlorophyll molecule is because it has electrons with the right energy level orbitals. And they will eject an electron. And the reason they eject an electron is because in these antenna regions of the photosystem, there are lots of different pigments embedded in those proteins. And they are absorbing all kinds of photons. And when they absorb the photon, as we learned earlier, the electrons will move to higher energy orbitals. And then that energy is passed from pigment to pigment. It's important to note that it is energy that's passed here in the light harvesting complex, these antenna molecules. It is not electrons being passed around. And that is important because there is not an electrical current going through here. It's just the energy that's being passed from one molecule to another. It's not until the reaction center that the electrons are lost by a molecule to go into the electron transport chain. Another way to think of it is this image that I found on the internet from another really good website, and it shows the energy as these dark arrows, and they show the photon as this yellow kind of lightning bolt. So the photon is absorbed, and then it is not electrons that are passed around, and it is just the energy itself. And then once all of that energy makes it to the reaction center, all of that energy together is enough energy for our chlorophyll molecule to actually lose electrons that then go into the electron transport chain. So this region of where our chlorophyll molecule is called the reaction center. And there's a reaction center chlorophyll molecule, and that is going to be either P700 or P680, depending on what photosystem it is. And then we have what's called the primary electron acceptor. And the chlorophyll molecule is not going to give the electrons directly to the electron transport chain. We have this primary electron acceptor that is going to make that transition between the photosystem and the electron transport chain. So questions on the test, definitely you're going to see a question about is it energy or is it electrons being passed around? When is it energy being passed around? When is it electrons being passed around? And always location, location, location. So these photosystems are embedded in the thylakoid membrane so that the active transport of protons are moving from the stroma into the lumen. So what's going in would be light, and then also water. So water will replace these electrons that get ejected from the reaction center. So we have water being used and consumed, and then products of just this particular part of the light reactions would be protons, and oxygen gas, and high energy electrons. The light harvesting complex is part of the photosystem, and it's the part of the photosystem that has all the antenna molecules or the Accessory pigments, so we've got a lot of molecules there that can absorb photons. P680 is the chlorophyll A molecule in photosystem 2, and P700 is the chlorophyll A molecule in the reaction center of photosystem 1. And photosystem 1 was discovered first, and it is a photosystem. It collects photons, and then it is second in the pathway of the light reactions. And electrons from photosystem 1 will be given to our electron carrier, whereas photosystem 2 was discovered second, but actually it shows up first in this chain of reactions. And the electrons that are ejected from photosystem 2 have energy directly from the photons that were absorbed. The electron transport chain is embedded in the thylakoid membrane. It is actively pumping protons into the lumen of the thylakoid. Oxygen is produced when water donates electrons to photosystem 2. CO2 is not used here. CO2 does not show up until the Calvin cycle in the stroma. Water, we do see water here. Water is being broken apart, it's giving its electrons to photosystem 2, and it's probably not really that it's giving it as much as photosystem 2 is taking it. And then the byproduct is oxygen. And then our electron carrier is here. Our electron carrier is in both parts of photosynthesis. The electron carriers are reduced in the light reactions, and they are oxidized during the Calvin cycle. And I have a link here. This is a group that is determining the structure of ATP synthase. So this is the accepted model of the structure and how ATP synthase works. It's pretty crazy. It turns and pumps. So if you have time, you might want to check that out. Next, we will watch a short animation of the Calvin cycle. So remember, the Calvin cycle is also called the C3 cycle. It is the second part of photosynthesis. It takes place in the stroma of the chloroplast. Don't confuse it with Calvin cycle. Don't confuse it with Krebs cycle, which happens in cellular respiration. Krebs cycle is also called the citric acid cycle and the TCA cycle. The citric acid cycle and the Calvin cycle are not the same thing. The Calvin cycle and the TCA cycle are not the same thing. The Calvin cycle and the Krebs cycle are not the same thing, but Calvin cycle and the C3 cycle, those are the same thing.
The Calvin cycle is a series of reactions that results in conversion of carbon dioxide into the organic molecules needed to build new cells. It occurs in the stroma, which is the area of the chloroplast surrounding the thylakoid membrane. During the Calvin cycle, carbon dioxide, CO2, is added to a 5-carbon molecule called RUBP. The resulting 6-carbon molecule is unstable and quickly splits into 2 3-carbon molecules called 3-phosphoglycerate. Using energy from ATP and reducing power from NADPH, which are products of the light reactions, the pair of 3-phosphoglycerates move through a series of reactions and are converted into 2 molecules of glyceraldehyde 3-phosphate. When several of these glyceraldehyde 3-phosphate molecules have been produced, some combine to make glucose, while others are reused in the Calvin cycle. To generate an entire new glucose molecule, the cycle has to turn several times, because each turn of the cycle adds only one carbon atom from each molecule of carbon dioxide.
McGraw-Hill makes a lot of really good animations. I know I've said this many times, but even in other science courses, once you're finished with this class, even into college and graduate school, if you find these short animations helpful, if you Google McGraw-Hill animation and then the topic you want to learn about, you'll probably find a lot of really good animations from them. So the Calvin cycle takes place in the stroma of the chloroplast, and it occurs in these 3 stages of carbon fixation, reduction, and regeneration. And as you saw in the video, carbon fixation, you do need to know the enzyme that is the catalyst for this reaction. It's called Rubisco. It is the most abundant enzyme on the planet, so it's very, very important. And a typical question for this part of the Calvin cycle would be to ask what the substrates are and what the products are for this particular reaction. So the substrates would be RUBP and carbon dioxide. And the carbon from carbon dioxide will be removed and added to RUBP. And that is the moment of carbon fixation, when the carbon from carbon dioxide forms a bond with RUBP, because now that carbon has been assimilated into an organic molecule, and now the organic molecule is bigger than it was before. So this puts more carbon into the organic material on the planet. So now it can be used to form one of the macromolecules. And as we discussed earlier, photosynthesis is important for 2 reasons. It gives organisms on the planet a new source of energy, and then also a new source of organic material, building material. And the first product of carbon fixation is very unstable and quickly breaks into 2 molecules of PGA. And then at this moment, when ATP and NADPH arrive from the light reactions, they are reducing agents, and they will be donating the high energy electrons from the light reactions. So this whole stage is called reduction because we have electrons being added to these molecules. At the end of phase 2, the products are G3P molecules, and they are also called PGAL molecules. They mean the same thing. It's an abbreviation for the same name. And what you need to know about this part of the Calvin cycle specifically is the fact that some of the G3P leaves the Calvin cycle to become glucose, sucrose, cellulose, a lot of different carbohydrates. So the actual product of the Calvin cycle is G3P, this 3-carbon sugar, not glucose. But glucose is commonly listed as the final product of photosynthesis, the whole system. And so a trick question would be to ask you, what is the final product of the Calvin cycle? And the correct answer would be G3P and not glucose. There are other steps between the Calvin cycle and a product that would be glucose. Something else you need to know is that a majority of the G3P that's produced gets put back into the Calvin cycle. And this stage is called regeneration of RUBP. And RUBP, remember, is the substrate for Rubisco. And carbon from carbon dioxide, in order for it to become organic, has to be added onto a molecule that's already organic. It needs a scaffolding. It's not the reaction that would occur to take carbon from carbon dioxide and then add hydrogens to it. That reaction would require so much energy that going through this cycle is energetically favorable for a plant. The plant would spend so much energy trying to build an organic molecule from scratch that the plant would not survive the process. It wouldn't be enough energy generated from even cellular respiration to help power that. So the reason that we need to regenerate RUBP is it is an energetically favorable molecule, and for Rubisco to use as a substrate to fix carbon. All right, questions for this material will be very similar to the ones that we've seen before. Where does it happen? Make sure you know that this happens in the stroma and not in the matrix and not in the cytoplasm. Once again, we look for carbon dioxide, oxygen, electron carriers, phosphorylation of ADP. And when we look back at the cycle, CO2 is being consumed. It's a starting material. And there's no O2, there's no oxygen gas present during this part of photosynthesis. Our electron carrier is present. The electron carrier here is going to be acting as a reducing agent. So our electron carrier is dropping off electrons. It becomes oxidized in the process, and then it can go back to the light reactions and pick up more electrons and hydrogen ions. We don't have phosphorylation of ATP here. We have dephosphorylation of ATP into ADP. So ATP that was generated during the light reactions are also acting as reducing agents and donating high energy electrons in those phosphate functional groups. It also can go back to the light reactions to be rephosphorylated. And remember, the phosphorylation of ADP in the light reactions is called photophosphorylation. And the phosphorylation of ADP that we saw in cellular respiration, that was substrate level phosphorylation and oxidative phosphorylation. And the rephosphorylation of ADP in photosynthesis looks a lot like oxidative phosphorylation in cellular respiration, but here in photosynthesis we have light also giving energy to the system, so it is called photophosphorylation. All right, knowing the input and output molecules for the Calvin cycle, so the reactants for carbon fixation would be RUBP and carbon dioxide. The enzyme is Rubisco, and then the products would be these PGA or 3 PGA molecules. And the second stage is reduction. It's called reduction because this is where electrons from the light reactions, which now are high energy because they were either generated from or contain the energy of the photons. And then the products of reduction are the products of the Calvin cycle, which is the G3P molecules. And because of the cycle, products of the cycle can be a tricky way to phrase that question. The products of reduction are the G3P sugars. The products of the Calvin cycle would be the G3P sugars, NADP+, and ADP. Those would all be considered products of the Calvin cycle because they are generated in the Calvin cycle. But the product, well, I guess the product of reduction is also ADP, NADP+, and the G3P sugars because they're all generated during that part of the Calvin cycle. And the last phase of the cycle is regenerating the starting material. So regenerating the RUBP. So RUBP would be a product, and then we also have ADP as a product as well. And the purpose of regenerating RUBP is to have the scaffolding to fix carbon, to take carbon that was in an inorganic molecule and put it into an organic molecule. And now living organisms can use that new carbon as a source for building the macromolecules. And then also remember that G3P is P-gal. Now let's look at all of photosynthesis one more time.
Plants to grow, they need inputs of carbon dioxide, water, and energy. The chemical process by which plants use these resources to manufacture glucose, the building blocks of plants, is called photosynthesis. In the process, oxygen gas is produced as a byproduct. The energy for photosynthesis originates in the sun and arrives at the earth as sunlight. This light has both a wave and a particle nature. The particles, or photons, are the smallest units of light. Photons oscillate along a path, which is measured as wavelengths. The light emitted from the sun contains photons in a wide spectrum of wavelengths, called the electromagnetic spectrum. Photosynthetic organisms use only a small portion of the electromagnetic spectrum, called visible light. Photosynthetic organisms contain pigments that facilitate the capture of wavelengths of light in the visible light range. The color of the pigment comes from the wavelengths of light reflected. Plants appear green because they reflect yellow and green wavelengths of light. Red and blue wavelengths of light are absorbed by these pigments and provide the energy that is used for photosynthesis. Within eukaryotic photosynthetic organisms, also known as photoautotrophs, the chemical reactions of photosynthesis occur within plant cells in specialized structures known as chloroplasts. Photosynthesis consists of two sets of reactions, the light-dependent reactions and the Calvin cycle. Within the chloroplast are small disc-like structures called thylakoids, which are surrounded by a fluid-filled space called the stroma. The reactions that synthesize glucose, the Calvin cycle, occur in the stroma. The light-dependent reactions occur in the thylakoid. It is here that conversion of light energy to chemical energy is initiated. In most photosynthetic organisms, thylakoids contain pairs of photosystems, called photosystem 1 and photosystem 2, that work in tandem to produce the energy that will later be used in the stroma to manufacture sugars. The photosystems of the thylakoid consist of a network of accessory pigment molecules and chlorophyll, the molecules that absorb the photons of light. Within the pigment molecules, the absorbed light energy excites electrons to a higher state. Photosystems will channel the excitation energy gathered by the pigment molecules to a reaction center chlorophyll molecule, which will then pass the electrons to a series of proteins located on the thylakoid membrane. Photons of light strike photosystems 1 and 2 simultaneously. We will examine what happens with the photons striking photosystem 2 first. The energized electrons are passed from the reaction center of photosystem 2 to an electron transport chain. The electrons lost by photosystem 2 are replaced by a process called photolysis, which involves the oxidation of a water molecule producing free electrons and oxygen gas. While this oxygen gas is a byproduct of photosynthesis, it is an important input to the cellular respiration pathways. As electrons pass through the electron transport chain, the energy from the electron is used to pump hydrogen ions from the stroma to the thylakoid, creating a concentration gradient. This gradient powers a protein called ATP synthase, which phosphorylates ADP to form ATP. The low-energy electrons leaving photosystem 2 are shuttled to photosystem 1. Within photosystem 1, low-energy electrons are re-energized and are passed through an electron transport chain where they are used to reduce the electron carrier NADP+ to NADPH. When the chloroplast is receiving a steady supply of photons, NADPH and ATP molecules are rapidly being provided to the metabolic pathways in the stroma. Therefore, the ATP and NADPH formed during the light-dependent reactions are used in the stroma to fuel the Calvin cycle reactions. The Calvin cycle consists of a series of reactions that reduce carbon dioxide to produce the carbohydrate glyceraldehyde 3-phosphate. The cycle consists of 3 steps, the first of which is carbon fixation. In this step, carbon dioxide is attached to ribulose 1,5-bisphosphate, resulting in a 6-carbon molecule that splits into 2 3-carbon molecules. The second step is a sequence of reactions using electrons from NADPH and some of the ATP to reduce carbon dioxide. In the final step, ribulose 1,5-bisphosphate is regenerated. For every 3 turns of the cycle, 5 molecules of glyceraldehyde 3-phosphate are used to reform 3 molecules of ribulose 1,5-bisphosphate. The remaining glyceraldehyde 3-phosphate is then used to make glucose, fatty acids, or glycerol. It takes 2 molecules of glyceraldehyde 3-phosphate to make 1 molecule of glucose phosphate. Thus, the Calvin cycle has to run 6 times to produce 1 molecule of glucose. These molecules can remove their phosphate and add fructose to form sucrose, the molecule plants use to transport carbohydrates throughout their system. Glucose phosphate is also the starting molecule for the synthesis of starch and cellulose. Plants produce sugars to use as storage molecules and structural components for their own benefit. By utilizing the energy of the sun, along with inputs of water and carbon dioxide, plants act as glucose factories.
All right, for these last couple minutes, I just want to go over some suggestions for your study guide, review, cheat sheet, etc. Some students can fit everything onto one piece of paper, and then that is really helpful. A lot of students, and I know for myself as well, once you've made that kind of a cheat sheet where you try to fit everything on the front page of one piece of paper, you really understand the material pretty well, and a lot of times you don't even need the cheat sheet. So even though this test is technically over just photosynthesis, you'll probably want to put, and I would highly recommend still putting some information about cellular respiration on your cheat sheet, more so that you don't confuse the two. You could have one set of notes that is just photosynthesis and then a completely separate set of information on cellular respiration, or you could put them side by side on one piece of paper so that you can quickly determine the differences between the two, which one is photosynthesis, which one is not photosynthesis. It just depends on personal preference. And I would have some kind of general overview image of cellular respiration somewhere. I would really recommend putting something like this right next to an image that shows all of photosynthesis, just for very quick references. For this test, there will be a portion that is on the electromagnetic spectrum, very general information. You won't have to identify any of the different ranges of wavelengths, but you do need to be able to look at the electromagnetic spectrum, and if you're given the wavelength, know if that means those waves have higher or lower energy compared to something else. You do need to know the Ableman Experiment, how, the setup, why he chose what he chose during the, what did he choose to use to represent photosynthesis? How was he able to track if photosynthesis occurred or not? If we made some changes to the setup, what kind of results would you expect? And then for photosynthesis information, very similar to what you need to know for cellular respiration. What are the stages? Where do they happen? Starting materials, products. Is carbon dioxide, oxygen, water, protons, electron carriers, ADP, ATP, where are all of those during the entire process? There will also be some information again on the redox types of reactions. What is reduction? What is oxidation? In photosynthesis, is it a series of reduction or oxidation reactions that are the driving force, the goal? So remember, cellular respiration was a lot of oxidation to get to the intended goal of energy removed from a molecule. Reduction reactions were still happening, but the goal was the oxidation of glucose. In photosynthesis, the goal is the reduction of carbon dioxide into glucose. Okay, now I'm not sure I said that correctly. I'm going to say it again. In cellular respiration, oxidation of glucose was the goal. Breaking those bonds open, catabolic reactions, exergonic reactions, and photosynthesis is the reduction of CO2. These are anabolic reactions. We are building molecules. They are endergonic. We are putting energy into molecules. And some structural pictures would be good so you don't mix up the stroma and the matrix, the cristae and the thylakoid space, intermembrane space and thylakoid space, thylakoid lumen. Make sure that you have those situated in your mind. Here are some examples of overview pictures of photosynthesis that I think are really good. The one in the bottom right is very detailed. I would probably use this one. However, for some of you, you just need the big ideas, and then one of these two might be better for you. And I have given some questions here, review questions. And then this is a short video going over similarities and differences between cellular respiration and photosynthesis. And just quickly, we will go through the answers for these. So the light reactions, those occur in photosynthesis. They occur at the beginning of photosynthesis. And photophosphorylation occurs here. So we have ADP being phosphorylated by ATP synthase, and it's being powered by photons. Oxidative phosphorylation occurs in cellular respiration. It is the phosphorylation of ADP by ATP synthase, but it is powered by the oxidation reactions of glycolysis and the citric acid cycle. So they're named for how they get the energy to do the phosphorylation. And oxidative phosphorylation takes place in the inner membrane of the mitochondria. The Calvin cycle occurs in the stroma of the chloroplast. The Krebs cycle takes place in the matrix of the mitochondria. The stroma is the cytoplasm of a chloroplast, and the matrix is the cytoplasm of a mitochondria. Stroma and stomata both are found in photosynthesis, but the stomata are the pores in the epidermis of plants that allows gas exchange, so let CO2 in and O2 out. The stroma is the cytoplasm of the chloroplast. The thylakoid lumen is the interior of the thylakoid membrane in chloroplasts. This is where proton concentration will increase during the light reactions, and this proton gradient will power ATP synthase. The inner membrane space is the space between the inner and outer membrane of the mitochondria. We do also have proton concentration increasing in this region during cellular respiration, and it will power the ATP synthase enzyme slash facilitate diffusion protein. It will power that in cellular respiration. When and where are the electron carriers reduced and oxidized? So in photosynthesis, the electron carrier is NADPH. It is reduced during the light reactions, and it is physically in the stroma when this happens. And the electron carrier is oxidized during the Calvin cycle during the reduction phase when it donates its electrons to the Calvin cycle. The proton gradients in photosynthesis are across the thylakoid membrane. There's a low proton concentration in the stroma. There's a high proton concentration in the thylakoid lumen. In cellular respiration, we had the proton gradient across the inner membrane of the mitochondria, high concentration of protons in the inner membrane space, low proton concentration in the matrix. ADP is phosphorylated during the Calvin cycle. It is phosphorylated by ATP synthase, which is embedded in the thylakoid membrane. And the ADP phosphorylation takes place in the stroma. Oxygen is used or produced during photosynthesis. It is actually produced during photosynthesis, and it is produced during the light reactions using photosystem 2, water splits called photolysis, and the water donates electrons to photosystem 2, to the chlorophyll molecule that is in the reaction center. Water donates electrons, and the byproduct is that oxygen is produced. So it would be produced technically in the during the light reactions. And oxygen is actually used during cellular respiration. It's the final electron acceptor, and then when it accepts those electrons at the end of the electron transport chain, it is reduced to water. Carbon dioxide is used or produced in photosynthesis during the Calvin cycle. It is actually consumed by the Calvin cycle. The carbon is removed by Rubisco and added to RUBP during the first stage of the Calvin cycle, which is called carbon fixation. Water is used or produced in photosynthesis during the light reactions. In photosystem 2, water will be split. It's hydrolysis or photolysis. Water is broken. You'll hear it called that. Water is donating electrons to the reaction center of photosystem 2, and the byproduct is oxygen. Energy is moving in electrons via the electron carriers. Electrons removed from water will be added to the chlorophyll molecule in the reaction center of photosystem 2. And then those electrons will gain all the energy from the light harvesting complex pigments. And then those electrons are ejected from the reaction center and then move through the electron transport chain, and the power will decrease of those electrons as they pump protons against their concentration gradient into the lumen of the thylakoid. Then photons that are absorbed by the light harvesting complex in photosystem 1 are going to also eject electrons from a chlorophyll molecule, and then these electrons that have moved through the electron transport chain will replace those electrons in photosystem 1. And molecules and atoms are accumulating inside the lumen of the thylakoid. We have oxygen accumulating also as water is being split and the electrons are added to the chlorophyll molecules. And we do have ADP accumulating and NADP plus accumulating at the Calvin cycle, but they can move to the light reactions where they will be rephosphorylated or pick up more high energy electrons. Water is consumed as it is donating electrons to the reaction center of photosystem 2. And we do have energy from photons being passed around in the photosystem in the light harvesting complex. Those are not electrons flowing, it is energy flowing. We have electrons disappearing from or being ejected from P700 and P680, and those are the chlorophyll A molecules that are in the reaction center of the photosystems. And if we look back at our picture of the light reactions, you see that P680 is the chlorophyll A molecule of photosystem 2, and P700 is the chlorophyll molecule in photosystem 1. Those are always very popular questions on this material. And the way you would notice molecules accumulating and disappearing would be pH changes, charge changes, the appearance of a gas, the disappearance of a gas, etc. And lastly, the Ableman experiment. Application questions for this material would be, what if he used a different source of white light? Would that change his results? If he used a different kind of bacteria, a different type of photosynthetic organism, how would that have changed the results? If he would have used filtered light instead of light from a prism, how would that change the results? And in general, for this material, knowing where you are is very important. If nothing else, if you have no idea what the answer is, take a step back and ask yourself, where am I? Am I in the lumen of the thylakoid? Am I in the stroma? Am I even in a chloroplast? And because specific reactions occur in specific locations, knowing the location where the question is being asked can help eliminate a lot of incorrect answers and can help you determine what the question is asking you and what the answer is. All right, good luck on the test, and let me know if you have any questions.